Good afternoon, good morning, good evening, depending on where you're connecting in from. My name is Paul Cotter. I'm a member of the Animal Microbiomes Working Group within the Phytobiome Alliance, and my main job is as the head of food biosciences in Teagasc in Ireland, and I'm also the head of microbiology in a company known as CSIC Biome. So welcome to our webinar series, and it's a real pleasure to have you here and to have two fantastic speakers to speak to us today. Before we get to the two presentations, I'm just going to tell you a little bit more about the International Alliance for Phytobiomes in case you're not familiar with it already. This is a non-profit consortium of industry, academic and governmental scientists, and its vision is that by 2050, all farmers will have the ability to use predictive and prescriptive analytics based on geophysical and biological conditions for determining the best combination of plants management practices and inputs for a specific field in a given year. It has a number of different alliance sponsors, which you can see on the screen there, and is focused on the complex systems that are present within plant-based agriculture. And so that ranges all the way from the soil to the plants and incorporates factors like the climate and weather and water and how they impact on microbiomes, as well as macroorganisms and macrofauna, and include arthropods as well as other animals and plants indeed. And all of these are obviously very much influenced by management practices, and so that's very much an area of interest. So within the Phytobiome Alliance, there are a number of different working groups, and these span regulation, microbiomes, controlled environmental agriculture, soil health. And the one that I have the pleasure of being part of is the Animal Microbiome Working Group. So within this working group, our aim is to identify knowledge and resource gaps to advance our understanding of the interconnections between people, animals, plants, and their shared environments as defined in the One Health approach. So we have a number of upcoming workshops. So these are the next events of note at the Plant and Animal Genome Conference in San Diego. And that's taking place in mid-January. So one workshop will be focused on exploring phytobiomes on the 11th, and on the 13th, there is a workshop relating to the uniqueness and commonalities between plant, animal, and soil microbiomes. So hopefully those of you that are attending that meeting will tag along and get to enjoy those. So today's webinar is being recorded and it will be posted on the Phytobiomes Alliance YouTube channels later today. So hopefully if there are particular aspects of it that you'd like to revisit or you'd like to draw to the attention of some of your colleagues, please do that. The presentation section, which will involve two 20-minute presentations, will be followed by a question and answer session. So for the questions and answers, I'll encourage you to submit your questions in the Q&A panel. For when questions are submitted, there is also the option for you to vote or select questions that you also like and think should be prioritized. And on the basis of that prioritization exercise, I will answer questions in that order. The chat panel isn't available for the attendees to use. It's just there so that you can see any messages that are forwarded by the organizers that might need to be put up as we go along. And it will be possible to download PDFs of the slides that are presented from the chat panel. There's also the opportunity to register to the mailing list to receive news about other events that are organized by the Phytobiome Alliance at the link that's provided below. So today, what we're focused on is presentations relating to understanding ruminant microbiomes to improve animal health, productivity, and the environmental sustainability of agriculture. And we have as I said, two fantastic presenters. Starting off with David. So David Yáñez-Ruiz from the Spanish National Research Council, or CSIC in Spain, to be followed by Sinead Waters from University of Galway here in Ireland. So I'm going to introduce both of them now rather than disrupting the flow later on. So starting with David. So David is a research professor, as noted in CSIC. He's previously worked in the University of Wales at Aberystwyth, and then subsequently at CSIRO and the University of Queensland in Australia. He leads a research group that's working on the modulation of the rumen microbiome to optimize ruminal fermentation, including nutritional interventions in early life and the assessment of the environmental impact of livestock production. He has participated in several EU projects, and I've had the pleasure of working with both him and Sinead as part of the Master Consortium. And he currently coordinates the Horizon Europe project, Re-Livestock, as well as being the coordinator of the Feed and Nutrition Network of the Livestock Research Group of the Global Research Alliance for Agricultural Greenhouse Gases. So David will be followed by Sinead, and Sinead is a principal investigator and lecturer in host microbiome interactions in the Environment School of Biological and Chemical Sciences, College of Science and Engineering at University of Galway, Ireland. She leads a research program that's focused on understanding ruminant microbiomes and their interactions with the host animal to improve animal health, productivity, and the environment, and is particularly interested in the role of the rumen microbiome, its interactions with the host ruminant, in order to improve nutrient utilization from feed and reducing greenhouse gas emissions. Her research is funded mainly by the EU Horizon programme, internationally and nationally by Research Ireland, as well as a number of different Irish agencies, such as the Department for Agriculture, Food and the Marine, and a number of other sources. She has many different international roles, such as the co-chair of the Livestock Research Group at the Global Research Alliance for Agricultural Greenhouse Gas Emissions, and is the Irish representative of the EU Expert Group on Agricultural Methane Emissions, appointed by DG Agri under the EU Methane Strategy to provide expertise to the EU Commission. I could speak at length about their very impressive resumes, but in the interest of time, I leave it there, and I pass on to David to begin his presentation. Okay, so we can see that, David, and you're off mute. Grace, over to you.
Okay, thank you, Paul, and thank you for the invitation to the Phytobiomes Alliance. So together with Sinead, we're trying to give you an overview of the rumen microbiome at different stages of life and how important they are for the health, for the productivity of the animal, but also for the sustainability of the whole livestock sector. So basically, I'm going to concentrate more on the early life stage of development of the rumen, and I will make some comments on the later life aspects of this early life stage, and then Sinead will follow with some interventions and some aspects of the rumen when it's more matured, and also some interventions that can be applied at different levels in relation to animal health and productivity. So, first, before we go into the actual development of the rumen, I would like just to spend a couple of minutes on just trying to describe the rumen for those who are not that familiar with this ecosystem. So the rumen is part of the digestive tract of ruminants, and you can see the three figures of three different approaches to digest feed. You have ruminants like a sheep, that is a herbivore, the same as a horse, that is also a herbivore, but the evolution went in different ways in the way they make use of the vegetables material. In ruminants, they develop a pre-gastric fermentation chamber with different microbes that we'll see later on, where most of the feed is degraded and fermented before it goes to the intestine, while in horses is different. The feed is degraded and fermented later on in the digestive tract. So in the rumen, the animal establishes a very interesting relationship with bacteria, archaea, protozoa, and fungi, that they degrade most of the nutrients that come with the diet, and they produce different byproducts that the animal uses as sources of energy and protein. So, apologies for that. So I was saying the rumen microbiota generates different products, and they can be used by the host as the main source of energy and protein. So most of the volatile fatty acid that is produced by these microbes is the main source of energy for the ruminant, while the protein component of these microbes is the main source of amino acids for these animals. If we could open a little hole here on the left-hand side of the animal with a microscope of the ruminants, we could see something like this. So it's a very active and dynamic and complex ecosystem of different microbes that are colonizing plant material, and they are degrading in different type of products. In this video, we can only see protozoa because they are large enough to be seen by an optical microscope, but there is more diversity inside with bacteria and archaea. This ecosystem has three main characteristics that are very important to understand the functions and also how to try to modulate. First, that is very redundant. So there are different species and groups of microbes that perform similar activities, so they can help each other in case there is any disruption of the activity. The ecosystem is also very resilient in the sense that it's very difficult to modify the composition and the activity, but also when we manage to modify that, if we stop the action that we are including in the rumen, then the rumen goes back to the normal or initial situation very quickly. And something that is really relevant, and it happens the same with other mammals and including humans, is that is host specific. So every animal, every individual has a specific microbiome, and this is very important to understand the variability. And as Sinead will present later, it's also very important in relation to genetic improvement and breeding. Ruminants have been, and of course the rumen microbiome has been with us for a long time. The first ruminants, as we see them now from the family Bovidae, appeared around 15-20 million years ago. And if you look at the scale in here, which is in millions of years, it was really recently when we started to domesticate those animals, first goats, then cows and sheep. And it was, we could say yesterday, when we changed the way we manage these animals in terms of nutrition, reproduction, etc. So the rumen microbiome has been with us for millions of years, and now we are obviously changing the way we relate with animals and keep livestock, and that has implications in relation to the health as well of the animal, but also the rumen microbiome. So Sinead will present more things about the adult rumen microbiome. I will focus now on the early stages of development of the rumen. A ruminant, so a calf, a kid, a lamb, when it's born, is not a ruminant. The rumen is still under development because they are fed basically on milk, and it requires some time for this part of the digestive tract to be fully developed. So at birth, the rumen is only 20-25% of the whole part of the pre-intestinal digestive tract, while then later on, when it's adult, is around 80%. And that requires a minimum around 8 weeks, so 2 months, where we can have like three main stages. The first one with no rumination, then a transition period where the animal starts eating some feed, and then rumination starts to develop, and then from 8 weeks onwards, the animal normally is weaned, and then rumination is becoming active. And in these three stages, we have two processes that occur in parallel and are equally important: the anatomical development of the rumen and also the microbial development. So I will go through the two of them to explain why they're important and why they're also interconnected, and how we can trying to help to optimize the development of the rumen in both cases. So if we look at the anatomical development, we are not talking about not only the growth of the organ as such, but also how the epithelium develops to maximize the absorption of those volatile fatty acids and different nutrients that are produced by the microbes. So here you have photos from the microscope on the epithelium of a rumen at week 1, 2, 3, 4, 5, 6, and you can see that as we move to the right and then to the bottom, then the epithelium starts to develop these papillae, basically to increase the surface of absorption. And we can do that basically by feeding not only milk, but mostly milk and concentrate that enhances the fermentation of microbes in the rumen during that period. So here you have three, on the right hand side, you have three photos of the rumen at 6 weeks of age of an animal that has received only milk and forage, but also milk and concentrate. You can see that those papillae are much more developed when there is milk and concentrate. That basically enhances the fermentation of microbes in the rumen during these first weeks of life. And you can see here, this is another experiment that we run here with calves from day 0 to day 55 of weaning. This is how the rumen grows in size in just 2 months. And the inside epithelium of a newborn animal, where the papillae are just starting to develop to a fully developed rumen on the right-hand side of a calf that has been weaned. And this is very important, because if the microbes are active but there is no surface to absorb the nutrients, then the animal is not maximizing the activity of those microbes. And I like to use an example. It's a very domestic example, but I hope you like it, to how we can develop a good epithelium. This is a photo of 2 different towels from Sarah Home website, and you can see on the left-hand side, this is a towel that is 25 euros, on the right is 17 euros. And the difference between a good and a bad towel, or a cheaper or more expensive towel, is basically the towel can make you dry quickly, so absorbs the water from your shower or your bath. And basically what we want to do with the rumen is to develop a towel that is closer to the one on the left-hand side. So fully develop papillae are more important to absorb the nutrients. How can we do that? Well, we can do that in maximizing the fermentation of microbes, but also we can add probiotics and yeast. A good example, because they produce butyrate, and butyrate is the main source of nutrients of the papillae. So when we add yeast probiotics in the diet of ruminants and in these stages of life, then the papillae they develop more quickly. And this is an example from an experiment that we carried out actually in the master project that was coordinated by Paul, using yeast probiotics in lambs during the first weeks of life. And we could see that in comparison with control animals, you can see the control in yellow, and in orange those that received yeast during the first weeks of life. The amount of beta-hydroxybutyrate, which is a metabolite that comes from the fermentation in the rumen in blood, at 7 weeks and 10 weeks of age was much greater in animals that received the yeast. And that was also correlated with a greater production of volatile fatty acids, as you can see here again at 6 weeks and 10 weeks of age. So before and after weaning, again with a significant increase on the amount of volatile fatty acids, so meaning more activity of the microbes in that developing rumen. Then we could see the same in the rumen, so greater concentration of butyrate, which is the main source of nutrients for the epithelial cells. And then we could see that the papillae in the rumen of those animals were much wider. So there was the surface to absorb nutrients produced by the microbes was much larger, and therefore then the result was a much healthier papillae in the rumen. These are photos of 2 rumens, one from the control animal and from the yeast animal, yeast-treated animals, where you could see the wall of the rumen inside. The papillae looks much more healthier. The one on the left-hand side is more it's called more keratinization, and the development of the papillae is not optimal. So again, it's not only about producing good fermentation, but it's also to developing the right epithelium to absorb those nutrients. This is just what I wanted to show quickly on the anatomical development. But if we go into the microbial development, this is something that happens really, really quickly. So from birth, and some works are also showing that before birth, what is the developing rumen is quickly colonized by microbes. And that colonization, as I said, occurs even before the rumen is developed. This is some results from an experiment that we ran recently here in Granada, where we slaughtered the calves at different ages of life from birth. So from the first day of life, day 0 to 3, 7, 14, until 20, until 55, when they were weaned. And you can see the concentration of both archaea and bacteria in the digester content on the epithelium, so associated to the epithelium in those animals. And you can see that even at day 0, day 3, even though the rumen is not developing, not fully developed, there are bacteria and archaea that are already present there. So the role that these microbes play in these early stages is really, really relevant, and it happens even before the feed is fermented and the rumination is becoming active. And the same we can say as well for all the parts of the digestive tract, like the cecum. So this is the same type of figure, but for the cecum, so post-gastric fermentation. I said at the beginning of the presentation that this is more important in horses, in equine, but it's also important in ruminants. Part of the feed is fermented in the hindgut. And there we could see as well that both archaea and bacteria were present right from birth in both the digestive content and also associated to the epithelium. So wherever intervention we want to apply on these processes of colonization, it has to be very early, because it happens also very, very early. And also something that we've been observing is that it's not only about the amount of microbes that are there, but also the type of microbes that colonize the rumen. There is a kind of a sequence of colonization. Here you can see the differences in different types of archaea, and we concentrated in archaea because, as Sinead will comment later, they produce methane, and there is a strong research effort focused on methane production. But you can see that there is a different type of populations from the mouth to the cecum, but also there is a kind of sequence also by age. So on day 0 and day 55, the populations shift to different proportional contributions. But what is important here is that those that are present on day 0 and 3, they are relevant later on in life. So those that are relevant for the fermentation of feeds, and in this case for the production of methane, they become important from the very first days of life. And this is an experiment that we conducted much earlier in our research group, but also emphasizes the need to modulate the rumen microbiome right from the beginning to really have a persistent effect. This is a trial that we conducted with goats that had twins, and those goats were treated with a compound that reduces methane, bromochloromethane, and then we treated one of the kids of the young animals with bromochloromethane, and the other was control, and the same we did with the control goats. And then when those animals were weaned, we measured methane production. As you can see here, those animals that were, so those kids that were treated, regardless of whether they were with the mother or not, they produced less methane, because this product was very active in reducing the activity of methanogenic archaea, which are the microbes that produce methane. Then we stopped the treatment, and then we measured methane 4 months later. And what we could see is that those that were treated early in life, but also were raised by mothers that were treated, were the only ones that remain lower in methane production. So suggesting that there was a kind of programming effect, but only in those that had direct in contact with the main source of colonization, in that case, which was the mother. And this is an area of research that has expanded quite a lot, and Sinead, myself, and other groups are also trying to develop in future research projects. The other aspect that is important here, because I mentioned the impact of the mother or not, is that whether the animal is raised under natural or artificial conditions. And this is very, very relevant when it comes to microbial colonization. We have compared the pattern of colonization in the developing rumen during the first weeks of life of animals that stay with the mother or are raised in artificial systems. And for those that are not that familiar with livestock production systems, most of the dairy systems, the newborn is taken away from the mother after birth, because the mother has to be milked, and it stays mostly in isolation or no contact with other adult animals during the pre-weaning period. And that is like stopping what nature has developed, which is a source of colonization from the mother from other adult animals, and that may have implications on a full colonization and development of the rumen. And we did that comparing twin kids that were, one was raised with the mother and the other was taken from the mother, as we do in dairy systems. And you can see here the pattern of colonization in terms of species. So those that were raised in natural systems with the mother, during the first month of life, they had a much greater colonization of microbes, and that was reflected in a much greater also production of volatile fatty acids as a result of the fermentation in the rumen. So that has led us to do more research on trying to develop strategies to compensate this lack of colonization and source of inoculation because of that separation directly from the mother. And this is just an example. It's obviously a very experimental approach, but also showed how maybe we can potentially manipulate in the future the process of colonization. Here we had 4 groups of young kids that were kept either as control or they were inoculated daily with rumen fluid taken from adult animals that were fed on forage or concentrate diets, and then a third group that was fed a mix of those 2 rumen fluids but autoclaved, just to inoculate the nutrients but not the microbes directly. And what we could see very briefly is that those animals that were inoculated with rumen fluid during the pre-weaning period, at weaning they had a much higher microbial diversity, much higher production of volatile fatty acids, and also the effect that happens at weaning when they don't grow as quickly as when they were fed on milk, because they have this transition between their milk and solid feed. They suffered less, so they recover much quickly in terms of body weight gain. So showing that, if we manage to develop a good microbiota during this period, maybe animals can face these early stages of development and the weaning process much better than others. So the idea is now to try to see if we can develop some kind of ruminal probiotics cocktail, ideally, but this is something that obviously will not happen in the near future. I think I covered this already, so I think I'm on time, so I will just conclude and then give the word to Sinead. So the idea is to just to show the importance of the microbiota in the digestive function and health, and Sinead will show more examples of that in the adult animal. The rumen developing stage, that window of time is very important, and it has short-term and long-term implications. The short-term are more related to the weaning period, which is a critical period of transition from milk to solid feed, but also has long-term implications because it may affect the potential of the animal's future in life, but also offer the potential to modify, using different interventions, the composition and the activity of the rumen later on in life. And with that, I'd like again to thank you for your attention. I'll be very happy to answer any questions later on, and I'll stop sharing. And it's all yours, Sinead.
Thank you, David. I'm just going to try and find my presentation now. No, that's not the right one. You see. Okay, can you see that? Is that okay?
Yes, yeah, all good.
Okay, great, thank you. Thank you very much, Paul, for the very nice introduction, and thank you to David for a really nice laying the foundation to what I'm going to talk about today. I'd like to really thank the International Phytobiomes Alliance for the very nice invitation to speak to you all today in relation to some of the research that we're doing on ruminant microbiomes, and particularly on their role in improving animal health and productivity, and of course environmental sustainability of ruminants and agriculture in general. So as Paul said, I'm based at the School of Biological and Chemical Sciences at the University of Galway and a member of the Rhine Institute. But I'm relatively new to the university, and before that I was based at Teagasc in Grange for nearly 19 years. And really a lot of the work I'm going to talk about today, was with colleagues in Teagasc, and were projects that we did there. So I'd like to acknowledge my collaborators from Teagasc. Okay. So in terms of outlining the presentation today, I'm going to talk about the role of microbiomes, particularly in the environment. So understanding the rumen microbiome to enhance the nutrient utilization from feed and to reduce methane emissions from agriculture. And that is critically important in terms of food security globally. So particularly, how can we use this rumen microbiome? We can use it particularly to develop new methane mitigation strategies, and also to understand the mode of action and how things like feed additives work and strategies like that. Another big role of the application of rumen microbiome data is in the improvement of the accuracy of selection for low methane emitting cattle. And we made great strides in this area in the EU project led by Paul and in collaboration with David in the master project. So I'm going to talk a bit about that research, and that has really led on to another EU project called Whole Ruminant. So in terms of that work, it's basically explained as well in a review paper that we wrote last year in the Journal of Dairy Science. Another area where we work in animal and ruminant microbiomes is the area of animal health. So moving outside the rumen microbiome to the area of nasal microbiomes, lung microbiomes, and the respiratory tract, just to understand more about diseases such as bovine respiratory disease, diarrhea in calves, and of course a new area of colostrum, and understanding, is there a microbiome in colostrum, what is it, and how can we use this to enhance animal health? So just starting with the rumen microbiome, in terms of global food security, ruminants are critically important. And we should not lose sight of that, because ruminants have a unique ability to convert plant material, which we cannot consume or digest as humans, and they convert that in their rumen into a high quality meat and milk protein for human consumption. So they have a massive nutrition value globally. They produce nutrient dense foods such as milk and meat for human consumption. And this is critically important for not just in developed world, but definitely in the developing countries. So the ruminants have a very specialized fermentation vat in what we call the rumen that can degrade and digest plant material, which we cannot digest, converting cellulose, lignin, hemicellulose into volatile fatty acids, which we can then use as an energy source to convert it into meat and milk products. So the relationship between the microbiome in the rumen and the host animal is a symbiotic relationship. It benefits both parties. So the rumen microbiome plays a key role in the fermentation of cellulose, hemicellulose, and fiber into products utilizable by the host. And they include short-chain fatty acids such as acetate, propionate, butyrate. And then the animal provides that substrate and the environment, that really key anaerobic environment for these microbes to thrive. So as part of this process, now David touched on the key microbes involved. These include prokaryotes such as bacteria and archaea. Archaea are the microbes that produce the methane, essentially. We also have eukaryotic microbiomes such as protozoa and anaerobic fungi. Anaerobic fungi are really critical for the production of enzymes that break down feed, and protozoa produce a lot of hydrogen during the fermentation process. During that fermentation process in the rumen, this is when during the breakdown of feed is when methane is produced, because a lot of hydrogen is produced in the rumen, which really that cannot be allowed to build up. As a result, the methanogens come in and they reduce the CO2 by forming methane in the rumen. So ruminant meat-derived methane is basically a very potent greenhouse gas. And this is the consequence of the very good work the rumen microbes do in the breakdown of feed and converting it into meat and milk. Unfortunately, methane emissions, which is a really important greenhouse gas due to its implications for global warming, that is a byproduct of that fermentation process. So basically, it has an atmospheric lifetime of around 9 to 12 years, which is relatively short compared to CO2 and other greenhouse gases. But ruminant methane contributes about 32% of global GHG emissions. And we're under international legislation due to, COP is on this week, so this is very timely. We are under international commitments to reduce agricultural greenhouse gases, including methane emissions, by significant amounts, 30% by 2030, according to the Global Methane Pledge. However, methane is part of the biogenic carbon cycle. So it's relatively short-lived. So when methane is produced and emitted from these animals, the methane, after around 12 years, through a process of hydroxyl oxidation, can be converted back into CO2. And during photosynthesis, this CO2 is consumed and goes back into plant material again, which is in turn then used by the animals. So it's part of this biogenic cycle. That's what makes it very different to fossil fuels. And there's essentially no new methane, no new greenhouse gases produced. So going back to the whole process of how is methane produced. First of all, it's belched out the front, so there's very little that goes through the other side. So it's not really farting methane; it's actually belching methane. So during the digestion of feed, when the rumen is essentially an anaerobic fermentation vat, it's around 80 liters, and basically this is a very unique environment where the feed and very complex carbohydrates can be digested down or broken down into acetate, propionate, and butyrate, which are volatile fatty acids which serve as the energy source for the animal. So during that process, hydrogen is used, electrons are transferred, and essentially hydrogen is built up in the rumen. What happens then is that the feed is broken down by fermentative anaerobes and primary and secondary fermenters, mainly the roles of the fungi, protozoa, and the bacteria. So to ensure that the hydrogen or dihydrogen doesn't build up in the rumen, the methanogens come in, they combine the hydrogen and the CO2, or they essentially reduce the CO2 to form methane. And methane is produced generally by a hydroneutrophic reduction of CO2 in the rumen. This is the main pathway by which methane is produced in the rumen. So hydrogen combined with CO2 and methane plus water. And this is carried out by methyl coenzyme reductase enzyme in the rumen. So there are other pathways to produce methane, such as methylotropic pathway and also the acetoclastic pathway. But this is the most important pathway. So how can we use this rumen microbiome data? We can use it in two ways. First of all, to help design methane mitigation feed additives, and also the elucidation of the mode of action of feed additives in rumen. These are just two examples on how we can use this information. First of all, you would have heard of Bovaer, which is a really important feed additive. It's approved for use in the EU and in other jurisdictions, in dairy animals in particular, to reduce methane emissions from agriculture. So it's a feed additive and it's very effective. It's a synthetic, non-toxic compound, and it reduces methane emissions by around 30%. The mode of action is very clear. It basically limits the last step in the methanogenesis cycle to the coenzyme M reductase enzyme, and essentially the 3-nitroxypropanol molecule was designed to be able to sit into the active site of this enzyme that catalyzes the last stage of methane production. And that really can give you an example, by understanding this pathway or the methanogenesis pathway, DSM were able to design this really effective feed additive that can reduce methane emissions by around 30% in cattle. The immediate reduction of methane once fed, it happens immediately, but the emissions will increase once you stop feeding the additive. So really we face a conundrum in trying to basically develop this for grazing systems. We actually tested this feed additive in Ireland as part of a paper by Kerwin et al. And you can see that, excuse me, that it can reduce methane emissions by around 30%. Excuse me. Another way we can look at the role of the rumen microbiome is in feed additives, is looking at the microbial community analysis after feeding the feed additives. We looked at another feed additive called calcium peroxide. This was part of the master project, and basically we saw a reduction of about 28% in methane emissions following feeding of this additive. There was no net gain in feed intake or average daily gain. And then we look at the particular microbes involved. We can see that there is basically little significant changes in the bacterial community for their alpha diversity, but there's very specific changes. The archaea, there was a significant concurrent reduction in archaeal and protozoal alpha diversity, which appears to be dose dependent, and an increase in alpha diversity of the fungal populations. So basically, this is a feed additive that really operates by changing the redox potential in the rumen. And we were looking here to see how does that impact the microbiome in these animals. More specifically, when we looked in much deeper, we could see there was more abundant lactic acid bacteria, such as Phytobiomes, Streptococcus, and Sharpea. And we know from other studies that basically lactic acid bacteria were increased in low methane emitting cattle and sheep. We also see that the archaeal community was dominated by Methanobrevibacter species, and there was a strong shift in the abundance of protozoal genera, and we see certain genera such as Polyplastron and others, Entidium, are more abundant in the treated samples, and others like Esotrichia are greater in the control samples. We can also use the microbiome to improve the accuracy of selection for low methane emitting animals. So breeding, as David mentioned, and others, basically animal breeding is one of our main strategies we're going to use in the future to try to mitigate methane emissions. And here in Ireland, we've been very lucky through the Master project, through the DAFM-funded Green Breed project, where there was a huge effort to measure methane and look at rumen microbiome from those animals in large numbers through the collaboration with the Irish Cattle Breeding Federation. And in turn, they were able to publish the first world first methane data for breeding bulls. So we have genetic evaluations now in place in Ireland for methane emissions. We know that inclusion of methane as an output in the breeding index is possible. We know that there's individual animal variation in methane emission, that this exists. And we know that it's heritable up to about 0.3 or 0.4 in certain cases. And good breeding decisions are cumulative and permanent. In studies in New Zealand, Suzanne Rowe's team, they can show that in sheep, methane emissions could be reduced by 10% after 3 generations. So you don't actually have, this will not have implications for grazing systems, it has loads of benefits. In Ireland, we have a genomic selection breeding program. So really this allowed us to build on that to integrate methane into the index. Looking at a study by Paul Smith in Teagasc, he was one of my previous PhD students. He collaborated very closely with the ICBF, where we were able to collect rumen samples and methane emissions data on large numbers of animals. With the first 300 set of animals, beef animals, we saw for the very high to the very low, there was a 30% difference in methane emissions between both of those. We carried out rumen mesh taxonomics on the 300 samples, and we found there was no great difference in terms of diversity or the overall shifts in the rumen microbiome. However, when we looked at very specific species, we could see in the low methane animals that there was an increase in lactic acid bacteria, particularly bacteria such as intestinal bacillum, Olsonella, and Sharpea, for example. There was also a difference in the archaea. We could see that the RO clade was increased, which are less methanogenic, as they've only got one copy of the MCR gene, and also some of the Methanifera genus, which is actually less methanogenic than Methanobrevibacter. So this is really interesting. We also saw similar trends with the volatile fatty acids, with an increase in propionate in those low methane emitting animals. We also went further to see, could we look at the microbial biomarkers as predictors for enteric methane emissions? And this is the work of Alan Kelly and Paul, which was presented at GGA when it was in Florida. And basically by adding data on the microbiome into a prediction equation with body weight and feed intake, for example, and weights like that, we could see that increased the prediction, the R-square we adjusted by when you added in Mogibacterium, intestinal bacterium, Pseudomonas, and the SGMT clade of the methanogens. So this showed us that we could actually try and integrate the microbiome data in the breeding program, that could be beneficial. We could see that the ruminal abundance of 3 bacterial genera in particular, and Methanobrevibacter, accounted for a further 21% in the variation in methane emissions. So moving further then to the whole, that was from the Master project, we moved to the whole ruminant project, which is led by INRA, and David and I are members of this project. And from the ruminomics project, which preceded it, they found that a core rumen microbiome could dictate dairy cow productivity and methane emissions. Since then, we're looking at human studies where we're looking at enterosignatures that define common bacterial gills in the microbiome, because as we know, the rumen microbiome data, when looking at particular taxa, it's very wide dimensionality, and it's very sparse data and difficult to analyze. As a result, looking at ecological gills as units of microbiome data reduction, this was a focus we had within the whole ruminant project. And this was led by the group in ERTA. So basically a guild is a group of microbial species that exploit the same class of environmental resources in a similar way. So essentially they have co-abundance partners and they're linked. And they mightn't even be very related, but they really they execute the same type of functionality. So the guild-based analysis was conducted on data from international studies. And this is from ecological relevant aggregation methods, identifying bacterial fungal groups and helping us to reduce dimensionality and sparsity of the data. So what we did is is an international approach carried out by Yáñez-Ruiz in ERTA. He led this project, and we had data sets from across different countries. So we had Holstein from Italy, UK, and France, Angus from Canada, from the Luguan study, Charlotte from Canada, and also the Kinsella composite group, and crossbred cattle. This is from our group, from Teagasc and ICBF, where there was 849 cattle contributed to this study. We carried out microbial community analysis in the individual countries, looking at bacteria, archaea, and protozoa, and then looked at the ruminant signatures. So basically carried out non-negative matrix factorization to reduce the down into the gills. So looking at a common abundance across these communities. We also combined this data with host genotype data and host phenotype data. So it was a complete set of about 2,500 data from 2,500 animals, which is quite large. We found that basically ruminant signatures such as UBA2, so this is a ruminant signature which is very like succinivibrio, was consistently negatively associated with methane emissions and positively associated with feed efficiency across all breeds and countries. And this is promising candidate for integration into breeding programs. We also found that there was breed-specific and common ruminant signatures capturing about 96 to 99% of the variance of all bacterial ecosystem. And also there was a lot of those, as I said, linked with methane emissions, feed efficiency. These were actually also under host genetic control using our SNP chip data, with heritability estimates ranging from 0.09 to 0.51. And looking just at the Irish data, I just extracted the Irish data from this, and we look that we can actually get really high heritabilities across these ruminant signatures. And these include ruminant signatures like, Ruminococcus, Succinivibrio, Sharpea, etc. So this is very exciting data. We're going to build on this in a project called MAGS in Ireland. We're going to integrate some of this data into the breeding program and select animals based on low methane emissions, and also as part of future whole ruminant projects. We're also involved in the Rumen Gateway project led by Sharon Hughes in Queens. And she's looking, this is a Global Research Alliance flagship, where we want to really get back into culturing. And we have this is a photo from my lab here in Galway, where we're doing anaerobic culturing and looking at, can we simulate the rumen environment and mimic unique rumen environment under the gas conditions. And we combine this with whole sequencing and multi-mass spec to get some confirmation on some of these species. We'll be looking at the diversity, ontogeny, and functionality of the rumen microbiome, combining it with sequencing. And this term really is looking at culturomics. So this is another part of the work that we're doing. And we're really looking as well at direct-fed microbials, this application for vaccines, etc. So moving on, the interest of time, I could talk about methane all day, but just looking at other microbiomes, the role of other ruminant microbiomes, and there's a big program funded by the past by DAFM in Ireland, which is for a US-Ireland grant, and with Whole Ruminant more recently. And this is in collaboration with Bernadette Early in Teagasc and students Stephanie O'Donoghue and Sabine Scully. So looking at respiratory, bovine respiratory disease or pneumonia in cattle, it's a really important disease. And this is a multifactorial syndrome caused by very complex interactions in cattle, and mainly young calves. And basically, it's a combination between the environment and pathogens and the host immunity that really impacts the host immune defense mechanisms and can result in development of clinical signs and secondary infections. So it's mainly a virus as the first infection, and then that's followed by a secondary bacterial infection after that. So it results in nasal discharge, coughing, appetite loss, elevated temperatures and depression, and it's very common in young calves. So both bacterial and viral pathogens can be responsible for disease onset. As I said, there's an initial viral infection leading to secondary bacterial infection. And respiratory disease was one of the most common amongst calves from under 6 months of age, essentially in Ireland, with around 40% of calves getting it. So this trend continues to 6 to 12 months of age, with up to 40% identified as having respiratory disease infection. So basically, some of the microbes responsible would be Mycoplasma bovis, Pasteurella, Mannheimi, and really Dave Johnson did a lot of work previous to Stephanie O'Donoghue on identifying some of these viruses and bacteria associated with bovine respiratory disease as part of a US Ireland grant. So BOHV1 is the one we were working on here in this project. It is large double-stranded DNA virus transmitted through mucosal secretions and can establish lifelong latency within the host. And we from some of the work we've been doing, it looks like the tonsil PGT is a possible latency site for this virus. And this is one of the studies that Stephanie completed as part of her PhD. She finished her PhD in July. She looked at characterizing the microbiota in the upper and lower respiratory tract following an experimental challenge. This was carried out with AFBI in Stormont in Northern Ireland with BOHV1 in dairy calves. So just looking at the design of the study, basically day 0 was a day of challenge. So the calves either got a PBS placebo or a BOHV1. And then we looked at clinical assessments, whole blood and nasal swabs throughout the experiment. And day 6 was when the animals were slaughtered. We looked at nasal swabs, pharyngeal, tonsil, and lung tissue. Now, we did a host of transcriptomic studies across these tissues, but today I'm just going to focus on the microbiome.
Sinead, can I just jump in maybe another minute or so?
Pardon?
We have about a minute or
oh,
two.
Sorry, yeah, no worries.
Okay.
And basically, just from this, I can skip through this, Paul, sorry. So basically, we found that there was really no difference in the abundance of genera between the controlled and challenged. But really what we're seeing here is that under favorable conditions, a real symbiosis exists between the respiratory microbiome, and we see it's quite consistent. But at times of stress or disease, a dysbiosis can occur, and that causes commensal bacteria to become opportunistic pathogens. So we see similar with subclinical bovine respiratory disease, and Sabine Scully has been doing really nice work in this area. And just to say, she's looking at area detection and microbes associated with subclinical bovine respiratory disease. And she was looking at nasal as well as the GI tract, and found that low abundant microbes such as Rhombustia and Butyrvibrio had a correlation with subclinical BRD. But we can see here, very interesting in this slide, is that the animals that did have some clinical BRD did also continue, if you look at their average daily gains here and the weights, that the ones that did have subclinical BRD were underweight even later in life up to gestation, which was an interesting finding. We also see that there's very significant correlations between the nasal and fecal microbiomes, which looks at the crosstalk and the pathways between microbiomes, particularly under times of stress. And we can see these interesting correlations here. You can read all about it in Sabine's paper. Also, I just mentioned diarrhea is another disease we've been studying, Sabine's work. And just to say, this is the paper that Sabine did, and it's similar to this when you look at when this event, day 21 here, was the diarrhea event. And you can see that there's been quite stability on day 7 and after the event on day 84. But we see a disruption or dysbiosis in the fecal microbiome, in this case, on day 21. And really, it looks as change in the relative abundance of microbes during diarrhea and confirms bacterial dysbiosis. And here's some of the microbes involved in that. But definitely Bifidobacterium were found in the healthy calves. The last thing I want to talk about is colostrum. This is work done through the whole ruminant project. We're interested in the colostrum as it's critical to calf health and development as a source of antibodies, nutrients, and growth factors. We have been looking, Sabine has been looking at the microbiome in colostrum collected from fresh and stored colostrum. So, farmers would typically store it in the fridge if they weren't able to get fresh colostrum for some of their calves at birth. And basically, we see that there is actually a microbiome in the colostrum. It's diverse and homogeneous. And we see a presence of Lactospirae is linked to quality and passive transfer of immunity. And colostrum management practices like storage can drastically alter, as you can see here, the fridge versus the dam for colostrum. And we are doing more work on metaproteomics and whole genome sequencing of this colostrum from cattle. And that's a paper that you can read from Sabine's work. And to conclude, Understanding the rumen microbiome is critically important if we want to reduce methane emissions and enhance feed efficiency in cattle. We see breed-specific, common heritable microbial gills associated with methane emissions and feed efficiency. We're making good progress in culturing ruminant microbiomes, particularly through the Rumen Gateway project, and knowledge of ruminant microbiomes is critical to improve animal health and diagnostics through development of therapeutics. And I just want to thank a lot of people, my colleagues, previous colleagues and collaborators at Teagasc, the University of Galway, Irish Cattle Breeding Federation, and IRTA, and our funders. And thank you.
Outstanding. Thanks ever so much, Sinead, and thanks to David before you. I really enjoyed that and very informative. Excellent work. Okay, so we have a few minutes for questions. I'm conscious people might need to leave at the top of the hour, so please proceed if you need to. We might go slightly over for a minute or two if we don't get through questions in that period, but let's see how we go. So I'm going to jump straight into it. I have a question, first of all, for David. So David, question from Amy is, why don't for the animals that are consuming milk and forage, why don't they develop papillae in the same way as those that are consuming milk and concentrate?
Yeah, that's a very good question. Thank you. Yeah, when the animal is fed on milk and then have access only to forage, the papillae develop, but they develop much lower. So basically, the fermentation rate and the degree of fermentation in the rumen is much slower and lower in the forage-fed animals. And then if we want to achieve the weaning stage much quicker, then is when we introduce concentrate in the feeding, because the concentrate includes starch and other components that ferment very quickly, and that provides the nutrients for the epithelial cells to grow, and that's why we use that. But yeah, if you keep animals on milk and forage, of course, as in nature, they develop also papillae, but it takes much longer.
Okay, thanks, David. For Sinead, so also from Amy, she was wondering, why do you think there's an increase in alpha diversity for the fungi that are receiving, I think it was calcium oxide?
Well, we don't know. So basically, we're looking into it in greater detail. There's change in the redox potential when you give the feed additive, so it could be something to do with that. So there could be, and also we're basically putting in, small levels of oxygen with this calcium peroxide potentially. So changing the redox potential could potentially do it. But also we are going to be looking at more specific. We have a whole genome sequencing and a metatranscriptomic data set that we're analyzing that could actually give us more information in that regard. But it's a good question, and we're asking the same ourselves. And I take your point here, Amy, as well. You're absolutely correct. It's the total global methane emissions. You're correct in that.
Right. Thanks, Sinead. Maybe back to David. So you mentioned the focus on archaea because they're mostly responsible for methane and carbon dioxide production. But it was mentioned that bacteria produces the substrate that they in turn use. Do you think the focus on archaea will be helpful in mitigating methane emissions?
Yeah, thank you for the question. That's very good. Yeah, we mainly focus on archaea because they produce methane and because they have a unique metabolic pathway to produce methane that is not that redundant as other metabolic pathways. I was talking about the redundancy of the microbial ecosystem. There are unique pathways that if they are inhibited or partly inhibited, then we can manage to decrease methane production. But yeah, the question is very interesting because we can also focus on previous stages on the fermentation when hydrogen is either produced or diverted to other metabolic routes, and that involves bacteria. And in that way, we can also reduce methane production by either reducing hydrogen production, modifying the overall fermentation pattern, or also promoting the growth of some bacteria that can divert hydrogen to other metabolic routes. So it's it could be a combination of both, and the main target is archaea, but of course bacteria are very important.
Okay, super. Sinead, I have a question for you myself. It's not in the chat, but I suppose it's important not to ignore the elephant in the room, and that there's a lot of controversy in the public media around Bovaer and conspiracy theorists and whatnot. So can you maybe provide some science?
Sure. Yeah, absolutely, and that's a really good question, and I've been asked that many times actually since it broke. So really, what happened? There was a lot of conspiracy theories that basically Bovaer could have very negative effects and even cause cancer. So basically, after Bovaer was used on farms in the UK and where milk was supplied to supermarkets, there was some negativity on Twitter, and there was images of people pouring milk down the sink because they were saying that it was a dangerous chemical, etc. So following on from that, we actually have to supply the science. So essentially, 3-NOP has been trialed. There's been trials for 15, 20 years on 3-NOP. It's only added to feed in very, very small amounts. It's very safe because it's broken down in the rumen and it's digested from the animal. And it's like a switch. Basically, once the Bovaer is ingested with the feed and it reacts with the archaea, as I said in the presentation, it basically docks into the enzyme active site of the MCR enzyme, and it stops methane production. So that we has gone through the EFSA approval process. In August, one of the trials was conducted there, where it was seen to reduce methane emissions by 30%, where there was no negative effects on animal health, animal performance. And that has been replicated across the world. That particular trial was in beef cattle, but it's been replicated, and there has been a successful EFSA approval for the application of Bovaer in dairy cattle and reproductive animals. So, and basically what they want to do now is get it approved for beef, and they're well on the way for that. So I would have no concerns about the safety of Bovaer when it's applied correctly in the manner that's recommended and properly mixed into the diet. So it is safety. And EFSA is really rigorous. Like, it took years to get that dossier over the line. And there's been trials in many, many jurisdictions across Europe, US, Canada, Australia. So I would have no concerns.
Great. Thanks, Sinead. So I'm very conscious of time. I'm maybe just going to ask one more question, because Aurel in the chat, in the question and answer, had a number of different questions. So I'm going to direct one to David. But for any of you who haven't had questions answered, both Sinead and David have agreed that please do contact them afterwards, and please email them if there's anything of particular urgency. So I'll maybe go with Aurel's first question, and it relates to early stage ruminants. And you said that they don't have a true rumen with no rumination in the absence of microbes, but that later on then you mentioned that microbes were present. I guess the answer is just over time the microbes become present then. Is that right, David?
Exactly. They become colonizing even though the rumen is not fully active, but they start colonizing the different sites of the rumen and the different metabolic niches. But yeah, the rumen is not fully functional, but the work is to colonize and start the fermentation slowly. So yeah, the rumen is not fully active, but the microbes are there.
Super. Okay. Thanks ever so much, David. Thanks to all of those in attendance. Please do follow up with any additional questions as mentioned. Thanks to the 88 participants who've continued right through the end. I think we had a max of about 113 or so at one stage during the presentation. Please do keep an eye out for the YouTube video that will be released later today, in case there's anything you want to hear about again or to direct onto your colleagues. A big, huge thanks to Sinead and David. Really fantastic presentations. I really enjoy them. It's great to have you on board, and hopefully the people involved will continue to want to engage with you and with the Phytobiomes Alliance to learn more about this topic. So thanks ever so much. All right.
Thanks, Paul.
Thank you.
Thank you.
Bye-bye.
Bye.